Bismillahirrahmanirrahim. Assalamu alaikum. Today my topic of discussion is spermatogenesis. This is a process which takes place inside the seminiferous tubules of the male testes. This process involves the formation of mature sperm cells or spermatozoa in the human male body. In this diagram or flowchart, you can have an access to general features of the spermatogenesis. This spermatogonium, which are actually the germ stem cells of the human male body, by the process of mitosis, they are converted into primary spermatocyte. These two cells, they both are diploid. Diploid means that they have two sets of chromosome. This primary spermatocyte, under the first meiotic division, will be converted into two daughter cells. These two daughter cells are known as the secondary spermatocyte. They both are haploid in number. Now, under the second meiotic division, the secondary spermatocytes are now converted into four haploid daughter cells. These daughter cells are known as the spermatids. These spermatids are actually immature and non-motile cells, so in order to make them able to fertilize the ovum in the future, they will be converted into mature sperm cells by a process of maturation or differentiation that is known as spermiogenesis. Alright, now coming towards the location of the spermatogenesis. The spermatogenesis takes place inside the seminiferous tubules of the human male testes. In order to understand this process, it is very much important to differentiate between the basal compartment and the adluminal compartment of the seminiferous tubule. Let's suppose that this is a cross section of the seminiferous tubule. This outer green color line is the basement membrane or the basal lamina. At the center, we have the lumen, and these cells are the neighboring Sertoli cells. You can see that I have drawn these blue color lines in between these cells. This represents the presence of the tight junctions or the zona occludens between these cells. As we know that the seminiferous tubules are made up of two types of cells. One is the Sertoli cells and the other is the Leydig cells. The Leydig cells are actually the interstitial cells. Our main concern will be with the Sertoli cells because the process of spermatogenesis is taking place in their neighborhood. So, the process of spermatogenesis takes place in a way that the first step takes place at the basement membrane and the second, third, fourth steps they now take place in the adluminal compartment so that the final sperms or the final sperm cells they will be produced and they will be directly thrown out into the lumen. So, the process of spermatogenesis takes place in this arrow direction. As I have drawn these arrows already, you can see that the whole process of spermatogenesis is taking place in this direction. So we have a final product that is the mature sperm cell that will directly go into the lumen. Now coming towards the focused diagram of Sertoli cells, let's suppose these are two neighboring Sertoli cells. These cells are also known as sustentacular cells because of the presence of these projections. I have already told you that neighboring Sertoli cells are joined together by means of zona occludens or tight junction. The presence of these tight junctions make actually the basis of the blood testis barrier. This barrier is very much important because it does not allow the already produced sperms from the lumen to go out into the circulating arteries outside the tubules. This dotted line represents the basal lamina and these are the Sertoli cells. Now, I'll tell you about the process of spermatogenesis that is taking place side by side. This spermatogonium, that is the stem germ cell, it is present under the shadow of these tight junctions. So, this spermatogonium will be present in the basal compartment of the seminiferous tubule. Under the process of mitosis, these spermatogonium are converted into primary spermatocyte. So, the primary spermatocyte will be thrown out or it will be produced in the adluminal compartment. Let me remind you again that the seminiferous tubule is divided into basal and adluminal compartments by means of these tight junctions which are present in between the Sertoli cells. These tight junctions demarcate the line between the adluminal and the basal compartment. This whole process takes place in a way that the final mature sperm cells are present near the lumen of the seminiferous tubule. So these sperm cells are directly thrown out into the lumen. In order to understand spermatogenesis in further detail, we should know about the spermatogonia. What are the spermatogonia? And the singular is spermatogonium. So, spermatogonium or gonia are actually undifferentiated diploid stem cells which will initiate the spermatogenesis at the time of puberty. Basically, what happens that in a human male embryo, the epiblast layer will give rise to primordial germ cells. These primordial germ cells will be fixed into the yolk sac wall. After that, what happens around fourth week of gestation, the primordial germ cells from the yolk sac wall will migrate towards the developing male gonads. At the end of the sixth week of gestation, these primordial germ cells will be completely fixed in the gonads of the male embryo and they will be inactive until and unless the puberty is initiated. So, the spermatogonia can be divided into three types. We have three types like type A dark, type A pale, and type B. The type A dark is a type of spermatogonia that has a very dark nucleus. So, under a microscope, you will differentiate the type A dark and type A pale on the basis of the color of the nuclei. These type A dark spermatogonia, that can be written as type A small d, these are actually the reserve cells. They are present in the basal compartment and they, under the process of mitosis or simple mitotic divisions, they produce two types of cells. One, they produce type A dark, similar to these, because they are the reserve cells, so they will again start the process of mitosis and they will produce more bulks of type A dark and type A pale. These type A pale are actually those cells of the spermatogonia category that will undergo active mitosis and they will produce further daughter cells. These daughter cells that will be produced by the type A pale are known as type B spermatogonia. Type B spermatogonia, under certain substances and chemical initiators, they will grow, they will mature, and after certain growth, they will be known as the primary spermatocyte. These primary spermatocytes are actually the types of cell that will undergo the process of first meiotic division, then it will produce secondary spermatocytes, and under the second meiotic division influence, they will produce four haploid daughter cells that are the spermatids. So basically, what happens that the conversion of the type A dark spermatogonia into type B spermatogonia takes place in the basal compartment of seminiferous tubule. Type A dark, that is produced by the first germ cell, they again go into near to the basal membrane, near to the basal lamina, and they start producing these two cells again and again so that the reserve of the spermatogonia doesn't deplete. After that, the type B spermatogonia will go into the adluminal compartment and it will give rise to the primary spermatocyte, and so on. The process goes on. The point that needs to be remembered is that the transit or the transmission of the type B spermatogonium into the adluminal compartment takes place by formation of new zona occludens tight junction behind it. It means that, let's suppose that this is the type B spermatogonium. So this now has to be converted into primary spermatocyte. So what will happen that this tight junction, it will be vanished and this tight junction will be formed behind this type B spermatogonium so that the blood testis barrier is not compromised at all. I hope so that you are now familiar to the basics of the anatomy of the seminiferous tubule and the basic concepts behind the spermatogenesis. Now I'll give you a small overview about how does the spermatogenesis initiates in the human male body. So basically what happens that at the time of puberty, the hypothalamus gets a signal and it starts producing the gonadotropic releasing hormone. This gonadotropic releasing hormone now acts on the anterior pituitary. So, under the influence of this hormone, the anterior pituitary produces its two hormones, which are the FSH, the follicle stimulating hormone, and the luteinizing hormone or LH. This follicle stimulating hormone will act on the Sertoli cells of the seminiferous tubule, and this LH will act on the Leydig cells. You can remind it by means of these letters, S for Sertoli and L for Leydig. These Leydig cells under the influence of LH will produce testosterone. And the Sertoli cells, as we have already discussed, that they are the major components or they provide the major neighborhood for the spermatogenesis. Up till now, we have discussed the spermatogenesis until the spermatids are formed. Now we'll talk about the differentiation or the spermiogenesis, which will produce mature sperm cells. You can easily see the difference between the shape. These spermatids, they are immotile, they have no locomotory organ, whereas if you see the shape of these mature sperm cells, they have proper tail or a flagella, they have a neck, and their head is also changed by reduction of the cytoplasm. Now, coming towards the last session of my topic, that is spermatogenesis or spermiogenesis, it is actually the maturation of the spermatids into motile mature sperm cells. Basically, the spermatids, the haploid spermatids, by the process of maturation of differentiation, will be converted into motile mature sperm cells, or also known as spermatozoa. This spermiogenesis is actually a combination of four basic steps. Number one is the nuclear condensation. The nucleus condenses and it becomes thickened so that it can give rise to a this shape of nucleus. Number two is the acrosome formation. As we already know that there are various organelles that are present in the cell, one of them is Golgi apparatus. So the Golgi apparatus of the spermatid actually give rise to a acrosome. Acrosome is a cap-like structure that is present near at the tip of the head of the mature sperm. It has certain enzymes, which we'll talk about in the topic of fertilization, which help in the decomposition of various substances of the ovum. Number third is the formation of a flagellum. Flagellum is actually a locomotory organ. You can see here, and it is extending like a tail, which helps in the motility of the mature sperm. And number fourth is the reduction of the cytoplasm. You can see over here that the spermatid, it is rounded in shape, it has a large variety, it has a large amount of cytoplasm, whereas here in the diagram of a mature sperm cell, the cytoplasm has been reduced, confined to the head, some portions in the neck, and a little portion extending around the flagellum. So now coming towards the diagram of the mature sperm cell, this is a mature sperm cell or a spermatozoa. It has been divided into three species: the head piece, the middle piece, and the end piece. The end piece, basically it is composed of the tail or a flagellum that is covered with a brown colored plasma membrane. And now coming towards the middle piece has the mitochondria covered with the plasma membrane, and the head piece or a principal piece. It has a nucleus, a centriole, and acrosome. This acrosome has enzymes which will help in the fertilization process. These centrioles, as we already know, that when this sperm will combine with the ovum, so we need cytoskeleton for the further meiotic divisions. So this centriole will help in that process. My next session will be of oogenesis in some detail. I hope so that you liked the video. Thank you.